To this point, everything that we've done has been laying the groundwork. So this is all stuff before this that you're going to need, and you're going to need to have at your fingertips. You're going to need to be conversant in these ideas. What we're doing from this point on, though, is going to be actual questions that are going to appear on your actual test. So we've laid enough groundwork just to make sure, just in case, I don't know, something went wrong at your high school and you didn't know some of these things. And yeah, let's go on and actually learn some interesting biology. Okay, so this is the last little bit of groundwork, but we need it for the end of this particular slide set, and at the end we actually have stuff that you are going to be tested on, so worth paying attention. All right, these are a couple of functional groups, and so by functional group I mean things that you'll see over and over again in atoms, and when you see them they will have particular properties. And we'll give them a particular name. These are, these are useful. And they're things that you probably would have picked up in chemistry class, but just from the point of view of making sure everybody's on the same page, let's talk about these. So the first one is a hydroxyl group, and it's got H and it's got O, the oxy part and the hydr part. Hydrogen attached to oxygen attached to something. So here's our little hydrogen, there's our big red oxygen, and attached to something. It's not a hydroxyl group if it's not attached to something. So this will be attached usually to a carbon, but it could be attached to all kinds of atoms. So for example, here's ethanol, good old ethanol down here. The stuff that you would call alcohol, this is actually just one of the many chemical things that can be called an alcohol. And those are things with an OH group, essentially, right there. Okay. Next one is going to be a carbonyl. You're going to find these in the bonds between proteins. There's going to be a carbonyl carbon, and that is going to be attached to a nitrogen. That's just how proteins work. And a carbon on the other side it'll be attached to. All right, and so yeah, if there was carbon either side, you could call this part a ketone. And if you could say there was if it's at the end of a carbon skeleton, then you could call it an aldehyde, but it's not. This is just a carbonyl because it's a generic case. And so shown here inside of acetone, there we have a little carbonyl carbon, and here in propanol we have a carbonyl carbon right there. So fine. Carboxyl group, which is C, double bonded to O. So we'll draw a double bond as two lines, and a single bond is going to be one line. And C, single bonded to O, H. Now you'll notice that oxygen here is always making two bonds. Oxygen makes two bonds to carbon. That's what makes it oxygen. And carbon, you'll notice, is always going to make four bonds. So 1, 2, 3, 4. There's going to be four lines coming out of it in some direction. Look down here. 1, 2, 3, 4. And this carbon has 1, 2, 3, 4. That tells you right away how many things have to be attached to each atom. This is just its property. And so carboxyl groups, yeah, look just like this. Okay, next one is an amino group. And amino groups can sometimes pick up an extra hydrogen ion, H+, and can then carry a positive charge. But right now we've just got a regular old amine, which is going to be NH2 right there, attached to something. So glycine, which is going to be one of the molecules that we can string, actually this is an amino acid, sorry, is going to have the amine group at one end, and look at this group at the other end. Do you remember what that is? We flipped it around left to right, but it's something we just covered. C, double bond O, OH is carboxyl. C, double bond O, OH. Doesn't matter which one is up, which one is down, whether they're on the right or left. We're just categorizing the four things that are coming out of this. We're saying one of them is something, probably a carbon, but the other three are going to be double bond to oxygen, single bond to OH. Yeah. And likewise over here, to be an amine, we're saying, all right, it's going to be a nitrogen atom with two hydrogens bonded and one bond going to something else that's not hydrogen. Okay, shown here is a sulfhydryl. There is an amino acid called cysteine that does have a sulfhydryl on it in its native state. And here it is down here. Yay! So on cysteine, we can see a couple functional groups. Look, NHH. Even though they're pointed down, which is a little weird, we aren't fooled, right? We know that nitrogen with two hydrogens attached to something else that's not a hydrogen, that's an amine group. That's easy. And look up at this end. Now we haven't seen it pointed up in the air yet. That's a little weird. But carbon in the middle, double bonded to oxygen, single bonded to OH, which is a hydroxyl group by itself, and then bonded to something else that's not a hydrogen down here. That's a carboxyl group. So yeah, we got it. So carboxyl on top, amine on the bottom, and sulfhydryl over here. And together the whole thing is called cysteine, the whole molecule. There you go. Hydrogen and sulfur. Yeah, the bond is going to be to the sulfur. And so sulfur is going to be happiest making two bonds like this, but there are other situations in which it can make more. Those are usually just found in chemistry, not as much in biology. This is the form that tends to pervade in biology. Phosphate, super important because phosphate is the thing on ATP, adenosine triphosphate, that is the energy currency of the cell. Yeah, you remember that stuff. Yeah, of course you do. And so what we've got here, this is our phosphate group. We're going to have phosphorus in the middle. Double bond up to an oxygen, or sideways or whatever. It doesn't matter what orientation. It matters what's attached. Yeah. And then two oxygens with negative charges over here, and another oxygen that is going to be attached to something. So if it's just a phosphate by itself, I think it's going to have a -3 charge, but if it's usually going to be stuck onto some other molecule, and so it'll be attached to something that is not hydrogen, essentially. Okay, ATP is actually adenosine, like the nucleotide, the A nucleotide, triphosphate. So it's going to have 1, 2, 3 phosphates attached to it. So it'll actually have 3 exact copies of this molecule. And they'll be joined up so that this oxygen is going to be attached to this phosphorus and also the next one. So they're going to be kind of linked up like that. And the way that ATP works is that we're going to attach one of those phosphates to something else, or we're going to break one of those phosphates off in order to transfer energy in such a way that it moves something and causes some other chemical reaction. You will see plenty of these as we go on in this class. But yeah, I just wanted you to know that ATP is regular old adenosine, the nucleotide that you find in RNA, and it's got 3 phosphates on it. And when we take away one of those phosphates, this is like, we would consider this like taking away its energy, although it's still got some energy. We would say it's now ADP, which is adenosine with 2 phosphates, adenosine diphosphate, which means 2 phosphates. So there you go. That's it. It's just a molecule in which energy can be stored and from which energy can be released. That's the idea. Okay, methyl group is carbon. 3 hydrogens attached to something that is not a hydrogen. Great. And if it was attached to 4 hydrogens, it would be methane, and that's a different beast altogether. It'd be coming out of a cow's bottom, I guess. Or out of a volcano, maybe. And so here's a methyl group, and they'll just show it as like CH3. That's like the lazy way to write this, because we know that carbon makes 4 bonds, and if 3 of them are bonds to hydrogen, then we don't really need to show it. We can just kind of say it like that, because yeah, you've already got a good picture because you know what atoms do and how they combine. Okay, there are enantiomers, which is right-handed and left-handed versions of these chemicals. So these 2 things up here are not identical, or rather you can rotate them around, but you can never rotate this one so that it will be exactly this. What you'd have to do is mirror it, like put a mirror up to it, and physically rearrange some of those bonds to get it from one to the other. There are enzymes that can actually rearrange the bonds without changing the chemical. They're called isomerases. Yeah, they can flip it from what they would call an L to a D. So L is the left-handed version, D is the right-handed version. And you can ask a chemist which, why the one is left-handed and the other is right-handed. There is a particular reason. It's based on a naming convention. But yeah, I think for our purposes, we just got to know that they exist. Now, interesting thing, all the sugars you're going to find in your body that you actually make and store and use are going to be D enantiomers. We do not mess with the L sugars. And all of the amino acids that are going to be in all of your proteins are going to be L amino acids. There are a few cases in nature where you can get L sugars and D amino acids. These are made at great energetic cost and usually for very special purposes by really weird bacteria. So they can be made. Physically, they can exist. But life as we know it does not use them generally. We use the D sugar and the L amino acid. And this is true of every bacterium, all the way up to animals like you and I. Okay, so one of the things that you are going to do, and that we are going to have a quiz on, oh my gosh, are we going to have a quiz on this, is going to be amino acids. And the reason why we're doing this is so we will be able to talk about proteins at the level of amino acids. So if I say it's got an arginine, I need you to know that arginine, oh, what's that? Or if I say it's got an R that's changed, that's got a methyl group attached to it, you gotta know what these are. So let's break these down. You've got your hydrophobic amino acids. And so these aren't completely hydrophobic, but we're talking about the side chain, the part that's different on every amino acid. And those are going to be more hydrophobic than not. Then you've got your aromatic ones. These are going to have a ring with electrons that can actually be in different places at different times. They can move around. So it's going to look like a hexagon with dotted lines around the middle of that, showing that it's got double bonds in there, but some of the electrons are kind of loose. And those are able to pair up and make bonds in different ways that you can't with some of the other ones. You got your basic amino acids, which are going to be lysine, arginine, and histidine. You've got your acidic amino acids, which are going to be aspartate and glutamate, which are D and E. And you got your polar amino acids. These are going to be ones that are hydrophilic, or are going to share electrons unevenly in that side chain. These are going to share electrons quite evenly in the side chain. And the aromatic ones are just, they're mostly even sharers. They're kind of hydrophobic, but they also do this other thing. So I want to remember them as aromatic. I think that's more useful. So these are the amino acids. And you're not going to have to learn the structure for them. That's maybe something you'll want to do later on, or at least have some idea of. But no, for this, what I need you to know is what's the name of the amino acid, what is the single letter code for the amino acid, and what is its property? What is one of its properties? So this is a good one to learn from. I would say make little flashcards of this. You are going to need to know this. This is going to come up not only on the quiz, but on your first test. It'll definitely be there. You will have to do something like half of these amino acids. So G is for glycine, A is for alanine, P is for proline, V is for valine, L is for leucine, I is isoleucine, and M is methionine. Most of those are easy because there's only one M amino acid, methionine, and it has the abbreviation M. So that works out well. Others are not so good. Like glycine, there's actually two other G's, glutamine and glutamate, but we'll figure out how to remember those. So I'd say you want to look online, look on YouTube, or browse around for good ways to memorize the amino acids and their single letter amino acid code. So phenylalanine, it starts with a PH, but it makes a F sound, an F sound, and so we're going to call that amino acid F, phenylalanine. Tyrosine, there's a lot that start with T, so we're going by the second letter there. And tryptophan, if you just say it like twiptophan instead of tryptophan, then you got it, and you'll remember that that's supposed to be the W. Yeah. So for a test, I would maybe give you the name proline, and you would give me the P and tell me it's hydrophobic. Or maybe I would give you the P, and you would say, P is for proline, which is hydrophobic. Yeah. So you're going to have to come up with the property, and I will either give you the letter or I will give you the word. Down here, lysine is going to be K. Now this is tough because you got lysine and leucine, which are kind of similar. Lysine is the really basic one, and leucine is just L, and it's nice and hydrophobic. So they're similar in the way their names sound when you pronounce them, but they're actually quite different amino acids. And so that's the one that you want to watch. Arginine is arginine, and histidine is an H. The two acids are going to be D and E because they're right after each other. Aspartate is, or aspartic acid is D, and glutamate or glutamic acid is another way to say it, is going to be E. And then the polar amino acids are going to be serine, S, threonine, T, cysteine, C, asparagine, we're going to go by the N right near the end, and glutamine, the cutamine, is Q. Glutamine, the cutamine. Doesn't have to be doesn't have to be something you tell anybody else, but if it works for you and it keeps it in your mind, then it's good enough. It's done the job. So yeah, come up with a way to remember those. That's the next thing we're going to be testing on, talking about, and sort of quizzing over during class. Yeah, so we will do that. Maybe I think I'll ask individual people at random to name one of these. Yeah, that's probably good. Okay, just so you know, ordinary amino acids are all put together by a thing called a ribosome. And a ribosome is fantastic. It assembles amino acids into proteins with just really great precision and speed. It's wonderful. There are a couple cases, though, where you can get proteins that are made, but they're not made by ribosomes. And these are made by things called non-ribosomal protein synthases. And so I just thought I would show you that these exist because they're kind of interesting and they yield some very useful drugs. A lot of antibiotics are made this way. An example would be a thing called vancomycin. And if you get a really bad bacterial infection, they will put you on intravenous vancomycin, which is a peptide down here at the bottom. So this is actually all protein down here. And it's got a sugar molecule attached to it. And this thing is actually built. It is actually built up of these amino acids. So first it's got a leucine amino acid. So that's just like normal. Then it's got a non-standard amino acid. And when you're putting them together one at a time, you can use non-standard amino acids. So it's possible to make amino acids that include other elements or would literally have any chemical property that you would want to include. And you can put these together with a peptide bond. So they would effectively be a protein, but they could be made of any amino acids you can dream up, rather. So shown here, this is a thing called a beta-hydroxytyrosine. And sure enough, it's like tyrosine, except there's an OH group right over here. That's neat. Shown down here, this is an asparagine, just the regular old asparagine. And then I forget what this one is called. There we go. Hydroxyphenylglycine. So it's not quite tyrosine, but it is kind of close. Hydroxyphenylglycine. Yeah. And great. So we got two of those in a row. And then we got a beta-hydroxytyrosine, and it's actually a beta-hydroxychlorotyrosine. You can see it's got this chlorine ion that's stuck on there. And so there's another one of those. And then the last amino acid, and it is an amino acid, just not a standard one, is a 3,5-dihydroxyphenylglycine, 3,5-DPG. And you hook them together in just this way, and you hook this sugar molecule on just so. And together, this is the antibiotic vancomycin. And yeah, in order to make this, bacteria have to encode all of these modules together. And they line up, all these proteins line up, and they will each transfer their little part of the final thing on there. So that's a different way to make protein, but it's not the normal way. Okay, some other just sort of normal molecules that you'll find in cells. So fats are made of, they're kind of a hybrid, like a chimera, or like a centaur, half horse, half man, why not? They're going to be a glycerol molecule, and they're going to have fatty acids on the end of it. So glycerol molecule looks like this. Anything that ends in OL is going to have hydroxyl groups on the end of it. And it's basically an alcohol. And over here you got your fatty acid, and look at all these carbons sharing with hydrogen. Carbon that's just sharing with carbon or hydrogen, that's called a hydrocarbon, and those are hydrophobic. These guys over here, anything that you have an oxygen in, is going to be hydrophilic. And so you got a molecule that's got a hydrophilic end and a hydrophobic end. When you hook 3 of these up, this is a triacyl glycerol, you're going to have a very nice hydrophilic end up here and a very hydrophobic end down here. And so this is going to determine the way these things pack. They're going to form little blobs with all of their hydrophobic ends together and all their hydrophilic ends pointed out into solution. Okay, have you heard of saturated and unsaturated fatty acids? I bet you have, like in cooking and stuff like that. And so if you're a fan of health and all that stuff, you'll know that unsaturated is the good kind, right? And you maybe even want it to be polyunsaturated. Saturated is the one that actually tastes delicious, but it doesn't do you much good. So the problem, or the difference between these two, is that saturated fatty acids are going to have a hydrogen on every possible spot on the carbon, so no double bonds anywhere. Whereas unsaturated ones, these two guys could be, instead of sharing electrons, they could each be holding on to a hydrogen with those electrons. But instead, they're sharing. So, all right, great. Saturated fatty acids, interestingly, tend to be solid at human body temperature, whereas unsaturated fatty acids tend to be liquid at body temperature. So monounsaturated is going to have 1 double bond. Polyunsaturated is going to have 2 or more double bonds. Saturated is going to have no double bonds. That's easy. Yeah, here's what a molecule looks like that actually makes up your membrane. So this is a phospholipid. And the ends here are just, see all those carbons in the center? That's the black molecules. The hydrogens are the little gray ones on the side. That's just a hydrocarbon. Those are fatty acid tails, but they're hooked up to a different hydrophilic group, and those are the kind that we end up putting in the membrane. So here we've got a saturated fatty acid, and here we've got one that's unsaturated, and when it's unsaturated, it puts a little bend in it. Yeah, and that's what makes these things not want to pack together and makes them stay liquid at human body temperature. So shown here, yeah, saturated lipids, if you only have saturated lipids, they will actually form a what's called a gel phase. It's almost like a solid, yeah, like a semi-solid. And they actually have a pretty high melting temperature. And so if you have these in your body or in your cells, they'll actually form little fat islands, like a little iceberg that'll float around the surface of a cell. And you can watch some of these in action on this really neat video. Watch them form up around the protein that is, I think it's an integrin that's going to unfold on the surface of the cell. If you have a mix of unsaturated and saturated, then the unsaturated ones actually keep the saturated ones apart, and they stop it from forming these little fatty icebergs, basically. So, yeah, just by the little kink in their tail from that extra double bond, they are what keeps our membranes fluid, because these hard gel membranes are very difficult to move around. So a cell is basically trapped. Its membrane is like a solid when it's got too much saturated fat in it. Okay, now let's look at some molecules, and these are the ones you're going to have to memorize. So here we got steroids and cholesterol, which is a steroid type molecule. And yeah, cholesterol is essential. We make a lot of it every day and transport it around. But when a lot of those transport vesicles crash because we've eaten too much of it, then it can contribute to cardiovascular disease, which is basically clogging of the arteries. So shown here is a nice little cholesterol molecule. Actually, no, is that? No, that's testosterone. Oh my gosh. Yeah, gotta read my own lecture. So this molecule is, no, that is cholesterol. So cholesterol is testosterone with a tail. Okay, so the central molecule here that you want to look for is this one. These 4 rings, like a messed up version of the Olympic rings with 3 6-membered rings and 1 5-member ring, together, anything that has that particular building block in it, that particular shape, is going to be a steroid of some sort. Now, cholesterol is going to be a steroid with this big long tail on the end of it. And it's going to be the only one that we learn that has a big tail like that. So if it is a steroid with a tail, you're going to be able to say, oh yeah, that's cholesterol. I'm not going to make you draw these from scratch, but I'm going to show you pictures of these, and you're going to have to tell me which is which. That is going to be one of the things that we do. And testosterone is a neat thing. Testosterone and estrogens, for that matter, are actually made from a common precursor. So androstenedione is a steroid, and actually all these steroids start out from cholesterol. So we're going to move these cholesterol, we're going to modify it in different ways, and we're going to turn it into a lot of different molecules that will eventually have different effects on the body. And so we can take this androstenedione and we can turn it into an estrone or directly into an estradiol. And once it's in the form estradiol, this is now one of the more potent, what we call estrogens. These are the things that will generally bias a human body to look like what you would consider a biological female. Down here we have testosterone, and there's a simple form of testosterone that's actually made, and it can actually convert back into androstenedione, so it can then get turned into estradiol. But there's another version of testosterone where we actually lock it in. And so if you have this enzyme called 5-alpha reductase, which is going to be turned on by genes that are encoded on the Y chromosome, that's going to lock testosterone in as dihydrotestosterone. And dihydrotestosterone is one of these molecules that once it's locked in, there's no good way for it to go back. So just like estradiol, once you have locked that in, that is now going to be a signaling molecule that will, generally be prevalent in biological females. But more on that in just a minute, because there's a lot of people out there. It's more a sliding scale between biologically female and biologically male. And it depends on the amount of these different things that you're making in different tissues and at different times. Yeah. Anyway, here's our two molecules that we want to look at and memorize. So you got your basic steroid base right in there, and then testosterone over here. You'll notice has these two molecules hanging up there. They're two methyl groups. And so I think of testosterone, I'm thinking of a raging bull or something like that. We're going to be very, very stereotypical if it helps us remember these things. So in this case, yeah, why not? And I'm meaning this not to say that this molecule is particularly manly or not. It is not. It is just chemistry. But if this helps it stick in your mind, then so be it. This is a great way to remember it. Or come up with a better way. And that's fine. You don't have to remember it this particular way. You just got to remember it. So you'll be able to recognize testosterone because it does not have a tail, but it has these two little methyl groups up there. So I think of those as like bull horns. So imagine a bull with these two horns on top charging at you, full of testosterone, just rage like mad, and it has no idea why. Yeah, that's going to be your testosterone here. And dihydrotestosterone is just this molecule, but we have added hydrogen here where this double bond was to sort of break that up, but it still has the two prongs, the two horns there. So that's still very recognizable. And if you said testosterone for either of these, I would allow it. I would say that's okay. Now, estradiol over here, our big estrogen, you see it's got one of those methyl groups, but it does not have the other one. So yeah, no two horns charging ahead here. Instead, you've got an OH group, a hydroxyl group, which I always imagine as every biological female that I met while I was dating just looking me up and down and saying, oh, just really not being all that impressed. And again, you can come up with other ways to remember it, that's fine, but if you see that hydroxyl over here, OH, and it's backwards, we can draw this molecule facing the other way like that. Then you know it's going to be an estradiol, an estrogen. It's not going to have those two horns on the top. So, how do we recognize those estrogens? Well, any of these molecules that has an OH down here and has one of these methyl groups up here is going to be a version of an estrogen. And estradiol is this particular version with the one hydroxyl group at the top. And estriol, which is a little bit weaker, is going to have two of these hydroxyl groups up here. The weakness or strength is going to depend on what molecules it binds to and what chemical reactions they cause. It's all very abstract and downstream. These molecules are just little pieces, like little puzzle pieces that slot into a particular protein. They don't have any particular powers on their own, but it's the whole network that they're part of. The protein that they're going to lock into, and the protein that's going to turn on, and the changes that's going to create. That's where the differences are going to come from in the effect of estrogen versus testosterone. Okay. And there's two others that you're going to learn because why the heck not? Yeah. So you got your cholesterol here. That's easy. We got a dog with a nice little upright tail here. Think of a little pup that's happy to see you, or maybe even a cat that's happy to see you, sticking its tail up in the air. Then you got your testosterone here with the two prongs going up there. You got your estradiol that's just, oh, at both ends. Just, oh. Again, more? Yes, more. Fine. Over here, you've got ergosterol. So, ergosterol is the version of cholesterol that fungi make and we don't make. This is a target, and when we want to go in and try to inhibit a fungal infection, this is one of the molecules that we can target because we don't need it. We don't have any way to make it. And so it's like cholesterol, but with kind of a bent tail, like something's gone wrong in the tail. That's the way I would recognize it. And it's got that double bond up there. So, cholesterol with, it's like starting to melt or something. That's ergosterol. You know something's wrong. You know we're in fungus town. It's not going to be an animal like us. And up here at the top, we've got winstrol. So this is one of the molecules that is a synthetic androgenic anabolic steroid. So synthetic, this is a thing that is made in a lab and does not occur naturally. Androgenic, it would bias you toward testosterone-like effects. Anabolic, in that it's going to lead to muscle growth. So this is when Barry Bonds or whoever is allegedly juicing, this is the kind of stuff they are allegedly juicing with. And it has all kinds of terrible effects on the heart, causes testicular shrinkage and all kinds of stuff like that, if I remember right. But yeah, you can see it is still the same basic molecule. It's still a steroid at the center, but it's going to have this extra 5-membered ring over here, which tells you, whoa, that's not like a regular normal steroid. This is going to be our synthetic one. So yeah, those are going to be the 5. Commit those to memory, or rather, don't commit the full structure to memory, unless you want to, that's fine. Commit the differences between them. These are the 5 you're going to have to differentiate. I'm going to give you one of these, and you're going to have to tell me which one I have just shown you. And yeah, that's going to be a little game that we play. So here we go. Here's to that game. Okay, and now, last little thing. So what's the difference between the effects of these things? So what you got here is a little tiny baby embryo, essentially. And fairly early on in the embryo, back when it still has a tail, there's a full-on tail there, and before it really has much in the way of limb buds, when they're just kind of little kind of nubs that are going to become arms and legs, you start out with the baby's little urogenital system. So this is the developing system. And whether this baby is going to turn out to be biologically female or male or something in between, they all actually start at the same place. And then the cumulative effects of that high 5-hydrotestosterone or of estradiol are going to eventually push it in a different direction. And so you're going to get this little starting format, and we're basically just going to rearrange the same parts. We're either going to fold this little glands inside, and we're going to make a what we would consider toward the female end of the spectrum, or we're going to seal up this little tube right here instead of having it as a hollow, and we're going to seal that up, and that's going to form the basically shaft of the penis, and the glands is going to be at the top. Either way, it's the same parts on the baby, and they're actually going to be, there's a point, I think it might actually be a couple weeks into the development of the baby, where you actually can't physically tell the difference because the genes that are going to determine which of these, or whether it's something in between, haven't really been turned on, haven't really had much of an effect yet. So that's kind of neat. And so it's if I remember right, it's something like 1 to 2% of people that are actually not fully at this end of the spectrum, not fully at that end of the spectrum, what you would consider biological male or female, but are actually somewhere in the middle, one of these intersex conditions. In some cases, they may look a little bit more like male or female. This may even be you or me, depending on I guess the way you know is if you have successfully reproduced and had a baby, then you're probably, yeah. some version of biologically male or female. But yeah, it's a lot of possibilities. And if you got a room with 50 people in it, chances are at least one of those people is something like this. So this is something that we don't really talk about, but it's actually really common and normal and just a part of biology. Yeah, there are all kinds of things that can happen. And it's all gradients, like how much of one thing you have at a particular time in a developing body. That's what determines kind of how it goes from there. So there we go. That's the end of that one. That's just a little interesting tidbit at the end. It's these molecules over here that I am going to hold you to knowing the difference between. And then molecules like this. I'm going to say things like hydroxyl later on. And you better make sure you know it. So if you need to make some flashcards just to go over those, I would say that's a good idea too. And then also the amino acids right on here. I'm going to give you either the name or the single letter amino acid code, and you're going to tell me the other one of those two things and its property based on this little chart right here. So thanks very much.